Good day, the grade 12s of life sciences. My name is Mr. Mutseira Mukunupi, and today we'll be going through the grade 12 paper 2 life sciences preparation for your final examinations. Now will be the last time maybe you see me, because now this will be your final paper of life sciences paper 2. Now basically the structure of the paper, if we break it down, it is total marks of 150, and the time allocated to finish writing the paper is 2 hours and 30 minutes. And the topics that are covered in paper 2 of life sciences are the DNA, the code of life, which covers 27 marks of the question paper, meiosis, which covers 12 marks, genetics and inheritance, which covers 45 marks of the question paper, and lastly evolution, that covers 66 marks of the question paper. Now meiosis was also covered in the paper 1, and the entire topic must be studied. There are no sections or parts that must be studied for paper 2 or for paper 1. The entire topic of meiosis must be studied thoroughly. Now, in previous reports of diagnostic reports nationally, there were mistakes that were done by candidates, and that led them in forfeiting marks and losing marks that were easily achievable. Now, one of the mistakes that was done is differentiating between the pairs of chromosome and the number of chromosome. Now sometimes a question is rephrased in a way that you have to indicate the number of chromosomes in a cell, or you have to indicate pairs of chromosomes. You know that the human somatic cell has a number of chromosomes of 46 chromosomes, but now if they are in pairs, we have 23 pairs of chromosomes. So those are one of the mistakes that was picked up from previous diagnostic reports. And another mistake that was done by candidates is when they state the natural structure of DNA. Many of them, they will write this double-stranded DNA, and we know that the natural shape of DNA is double helix DNA. It's helical in shape and is double-stranded. That must be indicated. And the differences between the pedigree diagram and the phylogenetic tree. There's a big difference between the two diagrams, even in their definitions. Well, in phylogenetic tree, we see the evolutionary relationships of organisms, how they've evolved to form one organism to the other, while in pedigree diagrams, we see a family tree, where we find parents which give birth to certain offsprings and so on. And now the theory of natural selection is one of the questions that are poorly answered by candidates. In a scenario where now the question is based on a specific example, candidates, they just write a general answer of natural selection, while instead of writing the answer in a specific type of question that is asked in the question paper. Another point is difficulty with the correct spelling of biological terms. We know that in life sciences, incorrect spelling may lead to a different biological term or change the entire meaning of the term. So it is very important that spelling is practiced and biological terms are studied thoroughly, so that no spelling mistakes may occur. The theory of natural selection and speciation. At times, the process of speciation is asked through natural selection. We know that the type of speciation that we cover in our grade 12 syllabus is speciation due to a geographical barrier, and that occurs through natural selection. In a question that speciation is asked through natural selection, many candidates, they write the theory of natural selection instead of writing the process of speciation, where new species are formed from the original one. Allopathic speciation is not accepted, as this one refers to the type of speciation. We only cover the general speciation through geographical barrier. Now some candidates, they tend to apply allopathic speciation as the answer if it is asked, but allopathic speciation is a type of speciation. So just be aware of that point. We don't write allopathic speciation. We're only dealing with speciation in general, and that occurs through natural selection. In our investigation question now, where now we have to indicate variables, our independent variables, dependent variables, and variables that are kept constant, and when we have to look at what can be done to increase reliability of an investigation or of the results, the validity of the results of the investigation. Now, sometimes the questions are phrased in different terms. For example, a question might indicate that what was done in the investigation to increase the reliability of the result versus to how the reliability of the investigation or the results of the investigation can be improved. The information might be given in a question paper. Now, the candidate, as a candidate, now you must go to the information that has been provided and indicate if whether the investigators or the scientists did certain things in order to improve the reliability. So now the answer must come from what was done in that investigation. This is different from a question that will be rephrased in a way that how can the results of the investigation be improved, or how can we increase the reliability of the investigation. So how the question is phrased, you must be very careful on that in applying your answer. One of the mistakes also done is the differences between an unreplicated chromosome and the chromosomes that has underwent replication, a chromatid and a daughter chromosome. Now, we all know that a chromosome is a single thread. A chromosome is a single thread like that, and if it undergoes replication, during interphase, we know that we double the chromatic network in the nucleus of the cell. Now, if this chromosome undergoes replication, now we have a replicated chromosome. Now, a replicated chromosome has two sister chromatids that are joined together by a centromere. Now, a replicated chromosome will be the one that has two sister chromatids that are joined together by a centromere. So this is also one of the mistakes that has been done by the candidates in previous years, and just want you to note of that, not to repeat the same mistake. Now, unreplicated chromosome, this refers to the chromosome as it appears before DNA replication takes place as a single thread. Now, a replicated chromosome, this one refers to the chromosome as it appears after DNA replication takes place. Because of DNA replication, all chromosome material is doubled. Hence, each replicated chromosome is made up of two chromatids that are joined together by a centromere. Now, a chromatid, this refers to each of two threads of replicated chromosome. And lastly, one of the mistakes that is done commonly by candidates is that they write daughter chromosome instead of a chromatid. Now, a daughter chromosome refers to each chromatid after it splits from its sister chromatid during anaphase 2 and moving towards the poles of the cell. Now we'll just cover a bit of a vision in relation to the DNA code of life. Now this is a question based on the DNA code of life. We know that DNA is made up of two strands that are double strand, and it is also made up of a sugar, a deoxyribose sugar, and a phosphate. Now the natural structure of DNA, as I've indicated before, it's a double helix, and we know that nucleic acids that we find in the nucleus of the cell, either DNA or RNA, they are made up of nucleic acid, nucleotides. Nucleotides, these are monomers, or we say they are building blocks of nucleic acid. And now, a nucleotide consists of three parts. It consists of a sugar portion, a sugar, and a nitrogenous base. Now, where do we find DNA in a cell? We find the DNA in the nucleus of the cell, and we also find DNA in the mitochondria that we term mitochondrial DNA. Now, a brief history in the discovery of DNA includes 4 individuals: Rosalind Franklin, Maurice Wilkins, James Watson, and Francis Crick. And Rosalind Franklin and Maurice Wilkins, they worked together on the X-ray pictures of DNA. And then James Watson and Francis Crick were also working on the structure of DNA at a different university. Now what happened is that Maurice Wilkins took the X-ray pictures that Rosalind Franklin took of DNA without a permission and gave the pictures to Watson and Crick. It was these X-ray pictures that helped Watson and Crick to establish the structure of double helix DNA. Now Wilkins, Watson, and Crick, they received a Nobel Prize for their work on DNA, but at that time Rosalind Franklin had already passed away, and she received a Nobel Prize after her death. Now this is a picture of James Watson and Francis Crick working on the structure of DNA, double helix DNA, and this is a picture of Rosalind Franklin, who took X-ray pictures of DNA. Now it is very important that you note that Rosalind Franklin did not discover the structure of double helix DNA. She only took pictures, X-ray pictures of double helix DNA, and it was those pictures that assisted James Watson and Francis Crick in discovering the structure of DNA. Now if you look at the structure of DNA, it is made up of 4 nitrogen bases, which are thymine, adenine, guanine, and cytosine. Now in DNA, because it's double-stranded, the nitrogen bases, they pair with each other. And adenine always pairs with thymine. You know that we find an apple in a tree, so we take the letters A and T, they pair together. And then in base pairing, guanine and cytosine, you know, we find the car in the garage, C pairs with G. And these nitrogenous bases are held together or joined together by a hydrogen bond. DNA also consists of a sugar portion, which is a deoxyribose sugar, and a phosphate portion. Now you can see the natural shape of DNA. It is double-stranded, it has 2 strands, and they are coiled together, or they are twisted to form a helix. Now if you see the base pairing there, thymine always pairs with adenine, and cytosine always pairs with guanine. And we know that the bond that joins 2 nitrogenous bases, adenine, the bond that joins 2 nitrogen bases, we call them hydrogen bond. And we also indicated that the DNA also consists of a phosphate portion, and the sugar, it is the deoxyribose sugar. The purpose of DNA, functions of DNA, is that they form genes that carry hereditary information, and DNA contains coded information for protein synthesis. Now moving on to DNA replication. Now in DNA replication, if we break down the term replication, now the term replica means exact copy. So this is a process where DNA makes exact copies of itself. And DNA replication occurs during interphase before a cell divides, either by mitosis or by meiosis. And then DNA replication occurs in the nucleus of the cell. How it occurs is that the double-stranded DNA unwinds, the weak hydrogen bonds break between nitrogenous bases, the DNA strands unzip, each strand serves as a template on which its complement is built. The free nucleotide of DNA build on each of the two strands by attaching their complementary nitrogenous bases. This results in two identical molecules. Each molecule consists of an original strand and a new strand. Now it is very important to note there's a huge difference between DNA replication and transcription that occurs during protein synthesis. In transcription is the synthesis of proteins, that is when proteins are made, and in transcription is when now the single-stranded mRNA is formed from double-stranded DNA. The two processes, yes, they include DNA, but they're different. While in DNA replication is when DNA makes exact copies of itself. Now, when the DNA strand unzips in DNA replication, both of the strands, they will serve as a template to build these identical DNA molecules. While in transcription is that a certain portion of DNA will unwind, and then it will form two strands, but only one strand will serve as a template where now an mRNA will be formed from that. So just make sure that you are able to distinguish between the two processes and not to combine the two processes together. Now, why is DNA replication important? It is that it doubles the genetic material so that it can be shared by the resulting daughter cells during cell division. It results in the formation of identical daughter cells during mitosis. Another nucleic acid that we are going to look at is the RNA, the location, the structure, and the function. mRNA is formed from DNA during transcription in the nucleus and functions on the ribosome during protein synthesis. We also have tRNA, which is located in the cytoplasm. RNA is a single-stranded molecule consisting of nucleotides. Therefore, there is no base pairing in RNA. Already there's a difference between now a DNA and RNA. We know that DNA is a double-stranded and now RNA is a single-stranded molecule. Each nucleotide is made up of sugar. Now the sugar in RNA is ribose sugar, and we know that the sugar in DNA is deoxyribose sugar. Phosphate portion and nitrogenous base. So those the nucleotides of RNA molecule consist of ribose sugar, phosphate portion, and nitrogen base. RNA also has 4 nitrogenous bases, which are adenine. Now, instead of thymine now in DNA, in RNA we have uracil, no thymine, cytosine, and guanine. And we have already indicated that there is no base pairing in RNA since it is a single-stranded molecule. Now, in protein synthesis, we look at transcription, which occurs in the nucleus of the cell. Now, the process of transcription occurs as follows. We have a double helix DNA that will unwind, and the double-stranded DNA will start to unzip when hydrogen bonds break to form two separate strands, but only one strand is used as a template to form mRNA. Now, using free RNA nucleotides from the nucleoplasm, the mRNA is complementary to DNA. mRNA now has coded message for protein synthesis. Once it has coded message for protein synthesis, the mRNA will now leave the nucleus to the cytoplasm, and it will now attach to a ribosome. Then the process of translation occurs. Now this occurs in the cytoplasm of the cell. Now each tRNA carries a specific amino acid. When the anticodon on tRNA matches the codon on mRNA, then tRNA brings the required amino acid to the ribosomes. Amino acids become attached by a peptide bond to form a required protein. Now, peptide bonds are type of bonds that join together two adjacent amino acids. It's very important in this part in translation that you don't confuse the anticodon that we find on the tRNA molecule and the codon that we find in mRNA. We know that anticodons are triplets of nitrogenous bases that are found on tRNA, and the codons are triplets of nitrogenous base that we find in mRNA. Now, if you maybe make an example of such, if maybe we have a DNA strand and then codons on mRNA and then an anticodon on tRNA. If maybe the strand is given as TCG, we know that during the process of transcription, now we'll have T and then we'll have A, C, and now we have G and AGC. That will be our codon and our anticodon. Since now in RNA we don't have thymine but we have uracil, and then we'll have cytosine and guanine. That will be like that during the process of translation. It is important to note that transcription occurs in the nucleus of the cell, while translation occurs in the cytoplasm of the cell, where now the mRNA now has the coded information and will be wrapped around the ribosome of the cell. There we have a simple diagram that indicates the process of protein synthesis. When now we have the double helix DNA. Now, as I indicated, the differences between now transcription and DNA replication. In DNA replication, the entire DNA molecule will unwind and will unzip. Weak hydrogen bonds break, and two strands will serve as templates. While in transcription is that a certain part or portion of DNA molecule will start to unwind and unzip when weak hydrogen bonds break. Now this portion will serve for the formation of mRNA. Now once mRNA has been formed, it now has coded information for protein synthesis to take place. Now a consecutive of 3 nitrogenous bases or a triplet of 3 nitrogenous bases on mRNA molecule is termed as a codon. Now in this example here of this mRNA molecule, we will have now 1, 2, 3 codons on the mRNA. And once mRNA has been formed, it will exit the nucleus via the nucleopause and gets attached to the ribosomes in the cytoplasm. And now the process of translation occurs. And now here we have the tRNA molecules. Now they will come to the cytoplasm to bring the amino acids to the ribosomes, and triplets of nitrogenous bases that we find on tRNA are termed anticodons, and they will match the codons on the mRNA molecule. And as they do that, the amino acids, they attach to each other, and they are joined together by a peptide bond. Now in DNA profiling, there are so many uses of DNA profiling. Now it can be used to solve crimes, to determine paternity. In organ donor, we see if the organs will be a match, and so on. Now it is very important. Later on we'll go and look how we can use DNA profiling to determine paternity. Since no individual has the same DNA except for identical twins, DNA profile is a process of determining an individual's DNA. Uses of DNA include solving crime, to confirm if people are related to each other or not, such as paternity. I'll just go through a few questions then, based on what we have covered. Now in question 1.1.1, it says it's a multiple choice question. In DNA molecule, guanine pairs with adenine, adenine pairs with thymine, cytosine pairs with adenine, or guanine pairs with thymine. We know that adenine always pairs with thymine, A always pairs with T, apple on a tree. One strand of DNA has 60 adenine molecules and it has 20 thymine molecules. How many adenine molecules are present in a double-stranded molecule? Now this is a very good question. Now this question indicates that in DNA, we know that DNA has 2 strands. It is double-stranded. And now it indicates also that one strand has 60 adenine and it also has 20 thymine. And what must be determined is that how many adenine molecules are there. Now we know that in the double-stranded molecule, the ratio of adenine to thymine is 1:1. Whenever there's adenine, there will be thymine, or wherever there's thymine, there will be adenine. So if you have 60 adenines, it means another strand will have 60 thymines. And if we have 20 thymines, it means we'll have 20 adenines. So now, how many adenines are there? You know, one strand will have 60 and the other strand will have 20. So our answer there will be C, which is 18. A molecule of mRNA is copied from DNA by a process of transcription. Our answer there will be A. A small section of mRNA has the following sequence on bases that codes for different amino acids. Which of the following is correct representation of the anticodons on the number of amino acid coded for by this section? Now we have been given an mRNA. Now we know that mRNA has, for this example, this has 3 codons. Now the question now, you need to determine the anticodons and the number of amino acids. Now we'll eliminate 9 because now we know that only 3 amino acids will be brought to the ribosome during translation. Now the task that you have to determine now is the anticodons. Now if we have G on the mRNA, then we'll have C. It will be C, G, A, G, C, A, U, U. So therefore our answer will be B. If 10% of bases in DNA molecule are adenine, what is the ratio of adenine to guanine? Percentages, it is out of 100. And we are told that 10% is adenine. Now, if 10% is adenine, that means thymine will also be 10%. But this is not what we are asked. We need to determine the ratio of guanine. Now we are left with 80% of bases. Now we are left with guanine and cytosine. And we know that the ratio of guanine to cytosine is 1:1. So now we have 80% of bases now. So half of 80% will be 40%. This means that guanine will be 40% and cytosine will also be 40%. Now we need to determine, we know that adenine is 10%, so guanine will be 40%, and if we work out the ratio, it will be 1:4. So our answer there is D. And now biological terms, as I've indicated in the beginning of the session, is that spelling mistakes occur where candidates now they provide wrong spellings to the biological terms, which changes the entire meaning of the definition. Nitrogenous base found in RNA molecule. Now this one says it is only found in RNA molecule and not in DNA molecule. Now it will be uracil. The sugar molecule found in nucleotide of DNA. You know that the sugar in DNA is a deoxyribose sugar. The organelle in the cytoplasm, which is the site of protein synthesis, that will be ribosome. And the bond that forms between two adjacent amino acids in the protein molecule, it's a peptide bond. And this is one of the mistakes that the candidates do. They write a polypeptide bond. That is not the correct answer. The correct answer is a peptide bond. The process by which DNA makes copies of itself, that is DNA replication, and the type of nucleic acid that carries a specific amino acid, that is the tRNA, and the organelle in which the cell where translation occurs in the cytoplasm of the cell. And now the item matching, we know that in this type of questions, this is where either you give your answer as A only or B only, both A and B, or none. Now the statement is the pairing of nitrogen bases. Now pairing of nitrogen bases occurs in A only, in DNA, A only. And then produce first X-ray pictures of DNA. We know that Rosalind Franklin was the scientist that took X-ray pictures, and Watson and Crick, they worked on the structure of DNA. Contains the sugar ribose, that's RNA molecule. Our answer is B only. Location of DNA, we find DNA in the nucleus and also the mitochondrion of the cell, so the answer there is both A and B. Now let's look at this question, which is based on a process that occurs in a protein synthesis. Now since we see there are ribosomes and tRNA molecules, we can immediately indicate that this is translation. And looking at the question, identify the process is translation. Identify organelle A, which is a ribosome, and molecule B, which is the mRNA molecule. The bond at D. Now we know that a peptide bond is the bond that joins two adjacent amino acid. Give only the letter of the molecule that carries an amino acid, is letter E. Now this is also one of the mistakes that candidates do. When a question asks for a letter, candidates, they give the name. So please don't do that mistake. Read the instructions very carefully. Is copied from DNA, you know it's B, the answer there. And then is the monomer or building blocks of proteins. It is C, the amino acids. Now remember, in this question, the instruction says you give only the letter. We're just gonna break for 5 minutes and then we're gonna look at genetics, just brief revision on genetics. So we'll just break for 5 minutes and then afterwards we'll go straight into genetics, and then we look at previous question papers and how we attempt in answering those questions. Thank you. Okay, now moving on into genetics and inheritance, let's jump right straight into it. Now genetics, we know the study of heredity and variations in living things. Now hereditary characteristics are transferred by gametes, which are the ovum and the sperm cell. Now during fertilization, we know that the male gamete, which is the sperm cell, from the father, and the female gamete, ovum, from the mother, they will fuse to form a diploid zygote from which new organism develops. Each organism inherits special characteristics from both parents that makes it very unique. So now this picture here shows examples of characteristics that we find in humans, whether a person has a cleft on the chin, a widow's peak, dimples, brown hair, hair color, blood group, brown eyes, unattached earlobe or attached earlobes. So basically those are very few examples of characteristics that we can see in humans and we term them the phenotypes. This picture is a picture of the father of genetics, which is Gregor Mendel. Now he contributed a lot in the study of genetics. Now he did experiments on tea plants and to show the heredity in the type of plants, and his work really did a tremendous contribution to the field of genetics. Now it's very important now we learn the terminology that we use in genetics. Some of these terms are often confused by candidates. The terminology includes a gene, which is a small portion in DNA coding for a particular characteristic. Alleles, these are alternative or different forms of a gene which occur at the same position of homologous chromosome. A genotype is a composition of a gene which has now two alleles for a specific trait. An organism's genotype is represented by two letters which represent the alleles. Each letter represents one allele of the gene that controls the trait. Homozygous is when two alleles of a gene pair for a particular trait on homologous chromosomes are the same. The two alleles can be both dominant, homozygous dominant, or recessive, homozygous recessive. And when we write them, the dominant we use a capital letter, and the recessive we use a small case letter. Dominant allele, an allele that is shown and or expressed in the phenotype when found in homozygous condition and heterozygous condition. A recessive allele is an allele that is not shown or it is masked in the phenotype when found in heterozygous condition. It is only expressed in the homozygous condition, and we can only see the phenotype of homozygous recessive only when it is in a homozygous condition. Heterozygous, two different alleles for a particular characteristic, and now usually when we write the genotype of heterozygous, we start with the capital letter and then the small letter. In a monohybrid cross, only one characteristic or a trait is being shown in the genetic cross. Now, Mendel's law of segregation states that during gamete formation or during meiosis, the pair of alleles will separate or segregate and randomly unite during fertilization. When sex cells are produced by meiosis, allele pairs they separate, leaving each cell with a single allele of each trait. Complete dominance, a genetic cross where the dominant allele masks or blocks the expression of a recessive allele in the heterozygous condition. In incomplete dominance, the genetic cross between two phenotypically different parents produces an offspring different from both parents, but with an intermediate phenotype. For an example now in this question, maybe you have a black fair coat color. Say the characteristic is a coat color. Maybe we have a black mouse and a white mouse, but the offspring of these two mice appears as gray. Now, a gray will be an intermediate phenotype between black and white. While in codominance, both alleles of a gene are equally dominant, whereby both alleles express themselves in the phenotype. Using the same example, we find the same parents of mice. We have a white one and a black one, but the offspring they appear as black and white. Now that's an example of a codominance. Now we have a white horse and a chestnut horse, but the offspring is wool. And now this will be an example of incomplete dominance. Now if we show the example of codominance, we have a red cow here, which is crossed with a white cow, and then we also have a phenotype that expresses equal proportion of the parents, which is wool. You see now we see both phenotype of the parents in the offspring. Now when we do monohybrid crosses, we use the following template or format to solve monohybrid genetic problems. We indicate the P1, and the P1 will state the parent generation, and then we indicate the phenotype of the parents, and then we also indicate the genotype of the parents, and then meiosis occurs. Now gametes are formed, then fertilization will occur. Now either for fertilization you cross the lines, or you provide an answer with a Punnett square. And then we indicate the F1 generation, we indicate the genotype of the F1 and also the phenotype of the F1. Now a genetic problem here, it says in jackals, red fair color, capital R is dominant over silver fair color. Do a genetic cross should a heterozygous red jackal be crossed with a silver? Now we indicate the P1 phenotype. We know the phenotype, we are crossing a red jackal with fair color. We're crossing it with a silver fair color. And it says it's a heterozygous red, capital R, small r, and the silver, you know, that is homozygous recessive. Meiosis occurs, and then gametes are formed. Now they separate. Fertilization occurs, where either we cross the lines or you write a Punnett square, and then we indicate the genotype of the F1 generation and the phenotype of the F1 generation. It is very important that in your answer you indicate the P1 and F1 generation and meiosis and fertilization, as this they carry 2 marks. A complete dominant example of a genetic cross which indicates a complete dominance is that one allele is dominant and the other is recessive, such that the effect of the recessive allele is masked by the dominant allele in a heterozygous condition. Now, in humans, the ability to hold the tongue is due to a dominant allele. A man heterozygous for tongue rolling and a woman who cannot hold her tongue have children. Use the symbol T and the small t for alleles of the tongue rolling characteristic and represent a genetic cross to determine the possibility or the possible genotype and phenotype of the children. It is indicated that the man is heterozygous and the woman cannot hold a tongue, which gives the woman's genotype as homozygous recessive. Now the answer will appear as follows. We know that the man is heterozygous for the condition and the woman is homozygous recessive for the condition. Meiosis and then fertilization, they must be indicated, including P1 and F1, as they carry 2 marks if they are indicated in the answer book. Then the gametes are formed by meiosis, and then fertilization occurs, and then now we can determine the genotype of F1 and also the phenotype of the F1 generation. An example of an incomplete dominance: a homozygous snapdragon plant with red flowers, capital R, was cross-pollinated with a homozygous snapdragon plant with white flowers. All the plants that grew from the cross had pink flowers. Represent a genetic cross to show the possible genotype and phenotype of the F1 generation of the plant. Now red flowers was crossed with white flowers and pink flowers were formed. Now this is an example of an incomplete dominance, where now the F1 generation will have an intermediate phenotype of the P1 generation. And now we use letters, capital letters, because now in incomplete dominance, there are no alleles that are dominant over the other one. Hence now we use different letters to indicate the genotype of a specific phenotype. And then meiosis occurs, which forms gametes, and then gametes are used for fertilization, and now we know that now all the F1 genotype will all be pink, which is the combination of each allele from each generation. In codominance also, a plant with white flowers was cross-pollinated with a plant with red flowers. All the plants that grew from the cross had flowers with equal distribution and red and white color. Represent the genetic cross to show the possible genotype and phenotype of F1 generation of plants. Still, we have alleles that are not dominant over the other one. Now we use different letters, capital letters, to indicate that they are dominant. Meiosis occurs, gametes are formed, fertilization occurs. The genotype is one of combination of the alleles, RW, and the phenotype now is that the flower will have equal distribution of red and white color. We know now this type of dominance is a codominance. Now in sex determination, we know that there's a 50% chances of having a boy or a girl. Now this is because in humans, somatic cell or diploid cells, 22 pairs of chromosomes, they are termed autosomes, 1 to 22 in a karyotype, and 1 pair of chromosomes, they are called the sex chromosomes or the gonosomes, which is pair 23. Now in pair 23 in males, they are represented as XY in males and as XX in females. The somatic cells of a male individuals, now they have 44 autosomes and 2 gonosomes, which are represented as X and Y. During meiosis, the homologous chromosomes pairs, they separate, and half of the male gametes will have 22 plus X chromosomes and the other half will have 22 plus Y. The somatic cell of females, they have 44 autosomes and 2 chromosomes, represented as XX chromosomes, with a total of 46 chromosomes. Now, in sex determination, when a female gamete, which has 22 and X, fuses with a male gamete with 22 plus X during fertilization, a zygote with 44 plus XX chromosome is formed, and this results in the development of a female. When a female gamete 22 plus X fuses with a male gamete 22 plus Y during fertilization, a zygote of 44 plus XY chromosome is formed, which results in the development of a male. Now, this is an example of a karyotype. Now, this karyotype is of a male because now the complete diploid set of chromosomes arranged according to their size and number in homologous pairs within a somatic organism. Now, this is a male karyotype. When we look at pair 23 in the karyotype, is that the chromosomes are not of the same size structure, which indicates X and Y. And then the female karyotype, when we look at pair 21, is that they have the same size, the same shape, the same structure. Now, we can easily distinguish between the male's karyotype and the female's karyotype. Now, representation of a genetic cause to show the inheritance of sex is that in the phenotype, the P1 generation phenotype, will have a male and a female. And then the genotype of a male is represented XXY, and the genotype of a female is represented as XX. Meiosis occurs, which forms the gametes, and then during fertilization, the genotype of the F1 generation is determined, and the phenotype, which is 50% female and 50% chances of having a male. Now, blood groups can be used to determine paternity. We know that in humans we have 4 blood groups which are controlled by 3 alleles. In humans we have 4 blood groups. Namely, we have blood group A, blood group B, blood group C, and blood group D. Now, I'm so sorry, we have blood group A, B, blood group AB, and blood group O. Sorry for that mistake. And these are the phenotype of blood groups. Now, if we determine the genotype of this phenotype, if a person has a blood group A, the genotype of a person with blood group A is capital I, A, capital I, A, or capital I, A, small I. A person who has a blood group B will have a genotype of O, IB, small I, and a person who has blood group AB will have a genotype of IA, O, and IB, while a person with blood group O will have recessive alleles to form that genotype. Now, the alleles that control blood groups, there are 3. We have capital IA, capital IB and small I. And the small I is the recessive allele, while A and B, they are both dominant. And if it occurs as AB, they occur as codominance because they are equally distributed in the offspring dominance. So blood group is controlled by codominance and multiple alleles. Now, we can use the blood groups to determine the paternity of a child. Now, how do we do that? The blood group of a child is determined by the alleles received from both parents. The blood group of the mother, the child, and the possible father is determined. Now, we say it's a possible father because we need to determine whether if he is the father or not. If the blood group of the mother and the possible father cannot lead to the blood group of the child, then the man is not the father. But if the blood group of the mother and the possible father can lead to the blood group of the child, the man might be the father, because this is not conclusive, as many men have the same blood group. And we can also use DNA profiling to determine the paternity of a child. And we do this by saying that a child received DNA from both parents. The DNA profiles of the mother, the child, and the possible father are determined. A comparison of DNA bands of the mother and the child is made. The remaining DNA bands are compared to the possible father's DNA bands, and if all the remaining bands of the child's profile match the possible father's DNA bands, then the possible father is the biological father. And if all the remaining bands of the child's profile does not match the possible father's DNA bands, then the possible father is not the biological father. If maybe I can do a sketch just to explain that one. For example, if maybe we have a DNA profile of a child, just gonna do a rough sketch to make an example. And then we have the mother, and the father. Now, what happens is that the child's DNA profile is determined, and also the DNA profile of the parents, both parents. And once the DNA profiles have been determined, is that we start with the DNA profile of the mother and the child, and we start to look at the bands that match, because she's the mother of the child. And now the remaining bands on the child's DNA profile will then be meshed to that of the father, and then of the possible father, and then if they match, just like the rough example I've made, then this man is definitely the father of the child. But if the remaining DNA bands on the child's DNA profile does not match any of the fathers, then this man is not the father of that child. Now, if we look at the questions that are based on DNA profile, that's like the questions here displayed. Now we have a diagram that shows a karyotype of a person with a certain disorder that is being caused by non-disjunction. And in our curriculum in grade 12, the disorder that we study that is caused by non-disjunction is Down syndrome. Where we know that now, instead of the pair 21 in the karyotype having 2 chromosomes, it now has an extra copy of chromosome that was caused by a phenomenon that we call non-disjunction, and it leads to a disorder that we call Down syndrome. Now, in non-disjunction, it's when the spindle fibers, they fail to contract and pull a chromosome towards the poles of the cell. Now, the disorder of this name is Down syndrome, because now we see on pair 21, we see 1, 2, 3 chromosome instead of 2. The phase of meiosis, when non-disjunction occurs, it can occur during anaphase 1, it can occur during anaphase 2. So the phase is anaphase 1 or anaphase 2. But the question indicated is meiosis, then anaphase will be credited. The type of mutation that result in non-disjunction, now this because it's a chromosomal aberration or chromosomal mutation. And identify the type of chromosome numbered 1 to 22. We know that the chromosome 1 to 22 are termed autosomes, and the chromosome pair 23 are termed gonosomes. Here we have an example of a species of fish has 3 phenotypes for fin length, elongated, short, and medium. Heterozygous fish have medium fins. So we are given 3 phenotypes there: elongated, short, and if they occur as heterozygous, they will have medium fins. Now already we know that if there is an intermediate phenotype, now this type of dominance is described as incomplete dominance. Now if we go to our exam guidelines, it clearly distinguishes between the 3 types of dominance. We have complete dominance, which is one allele is dominant and the other one is recessive, such as the effect of the recessive allele is masked by the dominant allele in heterozygous condition, while in incomplete dominant, none of the 2 alleles of a gene is dominant over the other one, resulting in an intermediate phenotype in heterozygous condition. So in our question there, we have an intermediate phenotype between elongated and short. The characteristic is under the control of one gene with 2 alleles. Elongated has capital E and short has letter S. So now if SS. Now the use a genetic cross percentage, two fish with medium fins having a offspring with short fins. So now the offspring that will have a medium fin will have one allele from each of the parents. So we'll have a genotype of ES. So this will be the case. We will have elongated fin, and this will be the phenotype that we are given, and we need to determine the genotype. Now if you do our genetic cross, remember we start with our P1 phenotype. Now the question indicates if you cross two medium fins, what are the percentage chances of having a medium fin? I'll be crossing two medium fins. Medium fin. The genotype is ES. We're crossing it with ES. Then meiosis occurs, which forms gametes. We have our E there, crossing. And now during fertilization, E will cross with each other, then E and S, E and S, and then S and S. These are our F1 generation genotype, and we need to determine the phenotype. Now if we have EE, we know it is elongated fin, we have one, and then we have two medium fin, and we have one short fin. Now if you go back to our question, we use a genetic cross to show percentage chance. Now it needs to be in percentages, so that it will be 25% chance of having a short fin when two medium fin fishes are crossed. Let's go to another question there. We have a black, the back of a leopard frog can be spotted as shown or be without spots. The spotted frogs are allowed to interbreed as they produce, and they produced 150 spotted offspring and 50 offspring without spots. Now, which phenotype is dominant? Is it the spotted frogs or the frogs without spots? Now, the answer there, it will be the spotted frogs, and the explanation is that when spotted frogs are crossed, they also produce frogs without spots. And now a frog that is heterozygous for spotted black was crossed with a frog without spots. Use a genetic cross. Now using the letters capital D and small d, represent the genetic cross to show the expected genotype and phenotype of F1 generation for 6 marks. Now it is very important in genetic crosses that you use the exact letters that are indicated in the question, as that when you use different letters that are not indicated in the question paper, he will not be credited marks for that. Now it is indicated that one of the parents is heterozygous for the condition of spotted. It's a spotted frog. It is crossed with a frog without spots. The genotype. Now it is indicated that the frog is heterozygous for the condition, the one that is spotted. So if it is heterozygous condition, then it will have capital D and a small d, and if it does not have spots, it will be small d, small d. Meiosis occurs, which forms gametes. And then gametes are used for fertilization when they fuse. And these will be our F1 generation genotype and the phenotype. We have 50% spotted frog and 50% without spots. It is important when we answer our questions on genetic crosses, remember P1 and F1 needs to be indicated, meiosis and fertilization needs to be indicated also. Here we have an example of a pedigree diagram question. Now, polycystic kidney disease (PKD) in dogs is caused by a dominant allele, and then the pedigree diagram below shows the offspring by 2 dogs. How many offsprings are affected by PKD? Now we can see if it is shaded, whether a circle or a square. Remember, in pedigree diagrams, a square represents a male. If it is shaded, it will be a male dog with PKD. And if it is shaded circle, it's a female dog with PKD. So we have 1, 2, 3, and 4 affected. Explain the genotype of offspring A. Shows that both parents are heterozygous. Now, if you look at offspring A, offspring A is a male dog without PKD. And we need to explain that the parents of A are heterozygous for the condition. Now, if you recall, the information that we are given is that the disease is caused by a dominant allele, which means now A received both recessive alleles from 1 and 2, which makes the genotype of A to be small h, and to be a male dog without PKD. We know that for each and every characteristic, we inherit one allele from each parent. So A inherited the recessive allele from 1 and another recessive allele from 2, which makes A to be a male dog without PKD. Now, if you do an example on a dihybrid cross, in a dihybrid cross, remember now we are looking at 2 characteristics at the same time. It is different from a monohybrid cross. In a monohybrid cross now, it's 1 trait at a time. Now, a certain plant species has the following alleles for each characteristic: number of seeds per pot, it is dominant 1 seed, and recessive is 3 seeds. Leaf shape, capital L is a normal shape, a small l is wrinkled shape. The table below shows the results of offspring produced by a genetic cross between two plants of this species. Phenotypes are one seed and wrinkled. You are given the information on the table there. And then how many genes of plant are considered here? Here there are two genes. Two genes. It is the number of seeds per pot and the leaf shape. So the answer there is 2. Name the dominant phenotype of the plant. You know the dominant one is capital letter, so it will be one seed and normal shape. Give the genotype of the parent. Now the parents, the number of springs are indicated. Now the genotype of each parent. Now here we have one seed and wrinkled leaves. So now if it is one seed, it will be capital P, wrinkled seed is small l, or it will be capital P, small l. If it is one seed, you know it's capital P, normal leaves will be capital L, or capital P, capital L, small l. Now the number of offspring that are homozygous recessive for both characteristics, the number of offspring that are homozygous for the offspring. Now if it is homozygous for both characteristics, needs to be 3 seeds and wrinkled shape. So it will be 32, our answer there. We have another example of a dihybrid cross, and then we'll do this example and then we'll look at evolution afterwards. And then we'll take just a small break after this one and then we deal with evolution. In holy trees, red fruit is dominant over white fruit, and spiny leaves are dominant over smooth leaves. A Punnett square below shows possible results of a cross between individual plants. Genotype at X is not given. Give the genotype at X, it will be capital R, capital R, capital L, capital L. The phenotype of the parents, we know that the information we are given is that red fruit is dominant over white fruit, and spiny leaves dominant over small leaves. And then what we are given here, we are given the gametes of the parents, and we see now these gametes of the parents are as a result of parents having characteristics that are both heterozygous for the conditions. So it will be capital R, small r, capital L, small l. Both parents are like that. And then here it requires a calculation in a population of 128 plants, how many plants with red fruit and smooth leaves are expected. Now what you do, you go to the table and then determine based on the information that you are given here, and then determine the calculation, how many are there, and then you divide by the total number of, which is 16, and then it will be let's determine this one. It will be how many plants with red fruit, and if it is red fruit and smooth leaves, it will be capital R and small l. So how many do we get there? It's 1, and then it's 2, so it will be 128 divided by 16 multiplied by 2. Then we'll get the answer. We just gonna break for 5 minutes, and then we look at questions that are based on evolution. We're looking at the theory of Lamarck, we're looking at the theory of Charles Darwin, natural selection process, where new species are formed, speciation. We look at human evolution out of Africa hypothesis. We look at bipedalism. We look at characteristics that we share with other primates and differences of characteristics with Homo sapiens and other primates. Now we're just going to take a 5 minutes break, and then we hit evolution afterwards. Now in evolution, since it covers 66 marks of our question paper, it's almost half of the question paper. Now this is where a lot of work needs to be done. The very first thing that we see from our examination guideline, we need to define biological evolution, differences between theory and hypothesis, and we need to go to evidence of evolution and differentiating between a biological species and a population, and factors or sources of variation, which include meiosis, crossing over, random arrangement and of chromosomes, mutations, random fertilization, and random mating. The theory of Lamarck, as it is used to explain evolution, Darwinism, and punctuated equilibrium. And while Lamarck's laws of use and disuse, law of inheritance of acquired characteristics were rejected, we need to know that. Now, when Charles Darwin explains the theory of natural selection, of evolution by natural selection, he says organisms produce a large number of offspring, and there's a great deal of variation amongst the offspring. Some they have favorable characteristics and some do not. When there's a change in the environment conditions or there is a competition, then organisms with characteristics which makes them more suitable, they'll survive, while organisms with unfavorable characteristics which makes them less suited, they will die. The organisms that survive, they reproduce and thus pass on the allele for the favorable characteristic to their offspring. The next generation will therefore have a higher proportion of individuals with favorable characteristics. In this way, the characteristics of a population gradually change over a long period of time. Now this is the generic way of answering Darwin's theory of evolution by natural selection. Now, in a case where this question has been phrased in a way that an example is given, now the answer has to be given in a specific way as it is asked in the question paper. You do not provide the generic the theory of natural selection to explain evolution. Now, if you look at this example here, there are two variations already. There's variation, and natural selection speaks of variation. There are two variations in the color of king snakes. Some have a bright, colorful pattern, and the other have a dull pattern. King snakes are non-poisonous to their predators. Coral snakes also have a bright color pattern, but are poisonous to their predators. This is a defense mechanism, as predators avoid them because they are poisonous. Scientists observed that king snakes shared the same habitat with coral snakes. There were more king snakes that had bright, colorful pattern. And the diagram below shows the distribution of the snakes. Now we have the bright king snakes, and from the information that we have been given is that the ones that the bright king snakes that are not poisonous, while the coral snakes are poisonous. And the king snakes that scientists observed that the king snakes share the same habitat with coral. There were more king snakes that had more bright blue colors. Now, they explain how the bright color pattern of coral snakes influences their survival. Now we know that coral snakes, they have bright color pattern and they are poisonous. So now the predators will not eat them. So the bright colors, what do? They make them, the predators, not to target them as prey, because now the predators know now that they have poisonous, or they are poisonous, so they avoid eating them. Now use the Darwin's theory of evolution through natural selection to explain why more brightly colored king snakes in this habitat. Now, as indicated in the examination guideline, this is the generic way of answering that question. Now we have to be specific on the question. We say now the snakes, they produce in a large number of springs, and there's a great deal of variations. Variation meaning there were king snakes that had bright colors, and there were king snakes that do not have bright colors. You know, those ones that have bright colors, they survived because now predators thought they were like coral snakes that were poisonous, and these ones that do not have bright colors, the predators ate them and they did not survive, they died. And these ones with bright colors, they were able to survive, and they were able to reproduce and pass on that allele for favorable characteristic to the next generation of offspring. Now, when answering this question on Charles Darwin on natural selection, it depends whether question has been provided within an example, or the question just states that just explain the theory of natural selection by Charles Darwin. Maybe we have more examples of this question here. Another example is this one, where now an ancestor of elephant had a long nose-like structure called proboscis. Each evolved a long trunk of elephant. The proboscis was used to gather leaves as food. The proboscis of Phiomia and the trunk of the elephant are shown in the diagrams have been drawn to scale. Explain the evolution of elephant's trunk in terms of Lamarckism and Darwinism, as well as the way in which the increase of length the trunk elephant could be achieved through artificial selection. Now this is a very good question. In this question now, you apply the theory of natural selection by Charles Darwin and to explain evolution, and the theory of Lamarckism. Remember now, Lamarck states that the law of use and disuse. If a certain body organ is kept being used frequently, then the longer or better it gets, or stronger it gets. Now, if you answer this question now, Lamarckism will state that the ancestral elephant stretched its proboscis frequently to get leaves on the trees, and then the more it used its proboscis, the longer it became. The offspring then inherited the acquired longer proboscis over many generations, and then the length of proboscis increased until it became a trunk. Now, Charles Darwin, when he explained how these elephants got a trunk is that there was a great deal of genetic variation. Some had long proboscis and some had short proboscis. There was a change in environmental conditions, and they had to reach higher in trees to get leaves. The animals with shorter proboscis, they died. Those individuals with longer proboscis, they survived. Then they reproduced and passed on the characteristics to the next generation of offspring. And the next generation of animals had a greater proportion and of animals with longer proboscis. Now in artificial selection, remember now, whenever the word artificial comes in, there is a human factor involved. Now humans, they want desirable characteristics from certain group of organisms to suit their own needs. Now humans select elephants with desired characteristics, and then they make them to produce offsprings with longer trunks. Those that are pure breeding are further selected to mate to produce offsprings with further long trunks. Now this question, we have to be specific on the example that was given in the question paper. Same as the theory of natural selection, the theory of speciation. Now, the speciation, as I explained before, it is due to a geographical barrier. Now, it states that if a population of a single species becomes separated by a geographical barrier, which can be a sea, a river, mountain, or lake, then the two populations, they split into two. There's no gene flow between the two populations. Since each population may be exposed to different environmental conditions, natural selection occurs independently in each of the two populations, such that individuals with two populations become different from each other, both genotypically and phenotypically. If the two populations were to mix again, then they will not be able to interbreed. The two populations are now different species. If you look at this question here, we have a diagram that shows a family of camels. We have Dromedaries, Bactrian, and a llama. And you know now they are of different species. Even if you were to mate them, they will not be able to produce a fertile offspring. Now this question is specific. Explain how speciation of camels may have occurred. Now, in our examination guideline, we have a generic way of answering that question, which can be asked in this type of fashion, just provide it this way. But because now this question has been phrased in a way that there is an example, then you have to indicate what organism is indicated in the question paper, and what is the geographical barrier that separates the population of those camels. Moving on, now we look at human evolution. In human evolution, we look at characteristics that we share with other primates, which include an upright posture, a large brain, long upper arms, eyes in the front or eyes with binocular vision or stereoscopic vision, eyes with cones, two mammary glands, freely rotating arms, elbow joints allowing rotation of the forearm, flat nails instead of claws, opposable thumb, which work opposite direction as the fingers, sexual dimorphism, rotate hands at least 180 degrees, olfactory brain centers reduced, parts of the brain that perceive information from the hand and eyes are enlarged. So these are the characteristics that Homo sapiens they share with other primates. But now when we look at differences between Homo sapiens and other primates, is that Homo sapiens they have a larger cranium to feed a large brain so that it can fit inside, which the advantage of having a large brain is that it increased intelligence, processing information better memory, higher intelligence, decision making. A flat face, while other primates they have a slopy face. Foramen magnum, more forward. Now having a more forward foramen magnum allows for the spinal cord to enter vertically, and now the Homo sapiens have ability to walk on 2 feet bipedalism, while the foramen magnum of other primates is more backward and they are quadrupedal. They do not have ability to walk on 2 feet. Bow ridges are not as pronounced, and row ridges are more pronounced in other primates, which always is involved with the diet, as now more pronounced bow ridges allows for large muscles for chewing and grinding uncooked food material. You can see also the Homo sapiens, they have smaller canines, as their diet is more of soft cooked food material, when other primates, ape-like beings, they have larger canines as they eat raw uncooked food, which they need to cut and slice with their larger canines. There's a smaller spaces between the teeth in Homo sapiens, while there's a larger space between the teeth in other primates. There are other characteristics, but we will move on. Now, if you look at the positioning of the foramen magnum in these 3 skulls here, you see that Homo sapiens, they have a more forward foramen magnum to allow the spinal cord to enter vertically into the brain and allow bipedalism. Also, the shape of the spine allows, which is S shape, which is allow flexibility, is also involved, is one of the characteristics that allows bipedalism in Homo sapiens. And the structure of the spine, which is shorter and wider to support the upper body, also is one of the characteristics that allows bipedalism in Homo sapiens. And the type of fossils that we find in the cradle of humankind, which belongs to Australopithecus africanus, the type of fossils include Littlefoot, Darwin child, Mrs. Pless. Now, those fossils they belong to the Australopithecus africanus species. You know that the Darwin child was found in the Northwest Province by Raymond Dart in 1924. Littlefoot, Australopithecus africanus, which was found by Ron Clark, and then in the Streepfontein caves, and Mrs. Pless by Robert Broom. And the Australopithecus africanus fossils examples, they include Karabo, Littlefoot, Darwin child, and Mrs. Pless. Now, when we come to evidence for that support the out of Africa hypothesis, out of Africa hypothesis indicates that all human beings arose from Africa, and they started migrating to other continents. And the evidence that supports this out of Africa hypothesis includes fossil evidence, as we know that the oldest Adipithecus fossils is only found in Africa, and then the oldest fossils of Australopithecus, which include Karabo, Littlefoot, Darwin child and Mrs. Bliss are only found in Africa. And fossils of Homo habilis are only found in Africa, and the oldest fossil of Homo erectus are found in Africa. And then also genetic evidence, which is the mitochondrial DNA, is used to support the out of Africa hypothesis. As you know that mitochondrial DNA is only passed by females. Now, the mutations that occurred in the mitochondrial DNA, when they are traced, they show that the oldest mutations was of a female that arose from Africa. Now, the grade 12s, it has been a great journey with you guys for the year of 2020. I know it has not been an easy year, and I want to wish you all the best of luck. I tried everything as possible just to cover some revision on paper 2 topics. All you have to do now is to prepare thoroughly, go over your work, use the examination guidelines to guide you on which topics are covered on the paper 2 examination. And I want to wish you all the best on your paper 2 life sciences. This will be your last time writing life sciences in grade 12, and the best of luck. And for me, Mr. Motsera Mukunupi, all the best. Thank you.